Any point, any questions or anything to clarify? Let me know. But I'm just going to start GI tract, finish that, then talk about hormones. That's how this is going to go. So, route of a taco well. Version 3.28154A. You have start at the mouth. You have your taco in your mouth. You're eating it. How does it get to your stomach? You start with your swallowing, which has two different phases or stages. Oropharyngeal stage, controlled by your swallowing center. In the oropharyngeal stage, mechanically, tongue pushes up in the bolus back into your esophagus. Uvula raises up to block your nasopharynx. Epiglottis also blocks your trachea because you don't want to choke on your food. As you're chewing and swallowing, you have different glands that are mixing saliva in with your food for both lubricative and digestive purposes. So your parotid glands will make up about 65% of your salival secretions. The mucus part of it, it has both mucus and serous secretions, mucus for lubrication and serous for digestion. Sublingual does about 5%, and it has only mucus for your lubrication, and then submandibular is about 30% of your secretions, only for serous, which is digestion. So, excuse me, you have your salivary amylase and your lingual lipase from your glands that mix with the food. As you swallow, you move into your esophageal stage, which is your peristalsis. Your peristalsis is an all or nothing reflex. So once you start it, it will continue all the way down the rest of the esophagus until it is finished. It does not stop partway. It is not a giver upper, it's a hard worker. You also have secondary contractions, which will help move a larger or sticky bolus down your esophagus as well. If we want to move on to, I will move on to histology, which is more the structure. And I think for the rest of this, I will talk more about structure before we go into function for the rest of the GI tract. So structure. Throughout your entire GI tract, you'll have a mucosal layer, a submucosal layer, and a muscle layer. Your mucosal layers will have a epithelium and then lamina propria, or your basement membrane, and then a muscularis mucosae layer. And then your submucosal will have various things, and then again, muscle is just muscle. So in the particular for the esophagus, you have your stratified squamous epithelium. And then in particular with the lamina propria, you have pegs of them in your mucosa. And then your muscularis mucosa in regards to esophagus is thicker in comparison to the rest of your GI tract. So as we go down to, and also, your muscle layers will change throughout your esophagus. And so as the proximal or the most distal part closer to your mouth will have more skeletal muscle. As you get to the middle esophagus, it'll be a mix. And then as you get down closer to the stomach, your muscle layers will become more smooth muscle. And so you get to your smooth muscle layer, you have a thick externa. As you get closer to the stomach, right where the gastroesophageal sphincter is, your stomach has different divisions. You have a cardia, which is, right before the sphincter of the stomach. You have your fundus, your body, which both are used for storage of food, and then your pyloric antrum and canal. In the pyloric antrum, the bottom part of your stomach is what does most of the mixing. In regards to the sphincter, as you do your peristalsis, that triggers the relaxation of the sphincter, which makes sense because if you're swallowing stuff, you want to be able to open that sphincter to let food into the stomach. That sphincter is there to prevent any reflux or any digestion of your esophagus by the stomach's acids. So talking about the histology of the stomach now, in regards to the mucosal layer, you have simple columnar epithelium. And then with the lamina propria is what carries the vasculature of the stomach. In the submucosal layer, you have a submucosal plexus, also known as the Meissner plexus. And then in the muscularis externa, you have three different muscle layers in the stomach. You have your innermost oblique, your inner circular, and your longitudinal. And between your inner circular and longitudinal, you have your myenteric plexus, also known as your Auerbach. Then also, I believe in your longitudinal, I forgot to write this here, you also have your interstitial cells of Cajal. And those interstitial cells of Cajal are neural cells that dictate the basal electric rhythm of your stomach, which dictates your contractions. And so if you look at the two different lines here, the bottom line is the level of activation by neurons, and then the top pattern is the contractions of your stomach. And so you have cholinergic and adrenergic stimulation of the stomach, and I believe that the cholinergic stimulation leads to stronger contractions, which makes sense because cholinergic is more parasympathetic, whereas adrenergic is more of a sympathetic. So you're probably not going to have a strong contraction stomach because you're not trying to digest. Neither confirmed nor denied yet, but that's just my speculation for now.
Say that last part again, what were you?
So cholinergic and adrenergic stimulation of the basal electric rhythm. So up here is pressure of the stomach or like the strength of the contractions. So cholinergic, being more, parasympathetic, will cause stronger contractions compared to adrenergic stimulation, which is weaker contractions.
Because it's vagal inputs versus the.
Like acetylcholine.
Yeah, acetylcholine being cholinergic, most likely to be parasympathetic, like you said, from vagus potentially, but I have not connected that to vagus. Again, going back into the the histology of the stomach. If we go to here, I've drawn a gastric gland where you have different types of secretory cells depending on where you are at in the gland. They're all scattered kind of throughout, but they tend to hang around their favorite area. So near the surface or like the opening of the gland, you have your surface mucus cells, which does alkaline mucus. And then at mucus neck cells, you have your acidic mucus that it secretes. Deeper in, you have parietal and chief cells, and parietal cells in particular are your important ones, secreting hydrochloric acid and then intrinsic factor. Your chief cells will secrete pepsinogen and gastric lipase, and then your G cells will secrete gastrin. In regards to the surface mucus, it makes sense. You're secreting alkaline mucus because you want to protect the lining of your stomach. As for the mucus neck cells, this one's an interesting one. I haven't really confirmed why you do acidic mucus, but my thinking is if you have alkaline mucus secreted at the top, maybe some of it leaks down into the gland itself. So you need the acidic mucus to protect the rest of the cells from that. But that's just speculation on my part and nothing I've confirmed. The question?
I'm just thinking out loud, and I don't want to disrupt your flow, so if you want me to.
No, feel free.
You have parietal cells and chief cells. Parietal cells are the ones releasing hydrochloric acid. Their proximity is directly below the mucus neck cells.
Correct.
So it might make sense that the reason you have acidic mucus in the mucus neck cells is because of their proximity to the parietal cells right below.
Potentially.
Because gastrin is the kind of mastermind, so it kind of starts at the bottom of gastrin. That releases parietal cells, and then they do the transition from pepsinogen to pepsin. Then you get the acidic mucus. So kind of like from the bottom.
Maybe. I like that actually.
Because what you just said, I had the exact same thought. It's like, why? How come there's acidic mucus over there? Because it's just kind of, that's one way I'm looking at it.
That does make sense actually. Yeah, because if you want to get that activation of pepsinogen, might as well like make sure you get as much of it as you can for
Right.
digestion. Okay.
But again, that's just spitballing. I don't know for sure.
I don't know either, but any ideas are appreciated, and I didn't think about it like that yet. Alrighty. I guess as I'm continuing, so I've gone over all the cell types of the lining of the stomach. They're not present throughout the entirety of the stomach. So if you go over to where I draw like colors on the stomach, representing the different cells, near the sphincter of the stomach, you only really have surface mucus and neck, which makes sense because you're not trying to digest at that part of the stomach, right? You're just trying to, close your sphincter, whereas the rest of the stomach, like the fundus body and the pylorus, you do want to have more of that secretion for digestion. So therefore, you'll have pretty much all the cell types. Something else to note, I didn't write it on here, but you also have C fibers kind of at the body and the fundus of your stomach, and those will sense heat and mechanical stimulus, and those can be used as what's the word? Referred pain, or used in the signals for referred pain, but that is not something I go into on this board at all. Also, in regards to the pylorus, you also have D cells aligning the stomach, which will release somatostatin, and I will be talking about all the inhibitory effects of somatostatin later. So getting all those out of the way, as you have your bolus go past your sphincter into your stomach, stays in the fundus and body where you have your solid chyme. In the body, in the fundus and the pylorus of your stomach, your pepsinogen is secreted from your chief cells, converted to pepsin by the acid. Pepsin also has an autocatalytic effect where it can also cleave off amino acids from pepsinogen to activate it as pepsin. And also, so that is partially, besides the HCLs, what's going to help digest your food. Your solid chyme becomes liquefied with the mixing from the pyloric antrum as it contracts. And there's peristalsis throughout the rest of the stomach to push that chyme through the pylorus where you have your pyloric sphincter. It doesn't, so of note, I only note that liquefied chyme will really go through the pyloric sphincter from lectures, but doesn't really explain why some solids make it through like corn. I speculated that, the liquefied chyme is still pretty thick and slushy, and so when you get into the intestine and it absorbs all the fluid, it consolidates it to stool, but again, it doesn't explain why there's solids that pass through. Maybe it's just once it just pushes it through regardless, but that's not explained in lecture directly. So as you have food in your stomach, there's a lot of regulation that can go on here. So actually, let me talk about the duodenum real quick because there's duodenal inputs in the regulation of movement of the stomach. And so duodenum is the first part of the small intestine where food will go into. And as food goes into the duodenum, it will carry some acid with it. So it'll drop the pH of the environment in the intestine, and that's one of the signals that affects gastric emptying. Other signals that affect gastric emptying is the presence of fats, the fluidity and volume of the chyme, and the stretch of the stomach. And so starting with just stretch of the stomach, your stretching of the stomach can just trigger release of acetylcholine. Signals can go to other parts of the stomach to release nitric oxide and VIP, and those as a result will cause the stomach to relax. So if you have food in there, the stomach will relax some more just to make sure you can eat more because otherwise it's contracted. The vagus, there's also signaling from the stretch to the vagus, and the vagus will also signal more release of NO and VIP, which is known as the vasovagal reflex to allow your stomach to hold more food. The chyme in the duodenum also will secrete ACH, cause the release of ACH to release more NO and VIP, and again, relaxation. And then, on the contrary, just to go back to that, if your stomach is empty and you're fasting, you release something called motilin. And motilin will trigger your migrating motility complex. And your migrating motility complex is just your rumblies you get when you're hungry. So that will start all the way at the stomach and continue down your small intestines. Now talking more about the duodenal input. So if you have, again, the decreasing pH of the duodenum, stomach distension. If you have fats in here, you also have proteins from food potentially. And so the proteins, they like to attract water because a lot of them have hydrophobic parts to it. And so that'll increase the duodenal hypertonicity, pull water towards it. Those will signal an inhibition of gastric emptying, which means your stomach will be contracting a lot less or pushing through food and chyme less in order to give yourself more time to digest. In particular, again, fat is the most potent inhibitor of gastric emptying because it takes the longest to digest out of proteins and carbohydrates. And so again, it makes sense you want to slow down your digestion, get as much out of your food as you can. Things that will speed up your gastric emptying is if you have pretty fluid chyme, you probably digested your food quite thoroughly, so you don't need to slow down your gastric emptying as much. The release of ghrelin, which is usually released when you're hungry, is also going to stimulate gastric emptying, although the reasoning for that, I have not figured out why that'd be important for that, unfortunately. What else am I missing here? Oh, so for stomach regulation as well, the decrease in pH will increase secretin. And that's just another way of saying, hey, in regards to lower pH, you don't want to digest your intestines. Secretin will increase secretion of NaHCO3, or bicarbonate, which will buffer the pH and protect your intestine. I haven't talked really about gastrin yet, and so once you're eating, your vagus is going to say, hey, I'm in rest and digest mode. I'm going to signal your parietal cells to release HCl. I'm also going to signal release of pepsinogen and gastrin. What gastrin will do is it will signal the release of pepsinogen from cheek cells as well. And then it also causes the release of histamine from enterochromaffin-like cells. And it's the histamine that will actually cause the release of HCl from the parietal cells as well. So you have multiple ways of signaling, hey, I want to release HCl, I want to release pepsinogen through vagal input and gastrin. As you eat, your D cells will secrete somatostatin. Your somatostatin will inhibit your ghrelin, so you're no longer getting the hunger signal. You're also going to inhibit gastrin, so you're going to need less of the digestive stuff at some point because you can't just have too much feed forward signaling. You need something to act, like regulate and inhibit it.
You go over that last part one more time.
Somatostatin inhibits ghrelin, inhibits hunger, so you won't eat less. It also inhibits gastrin to some level, which, because I said it makes sense to have something controlling this instead of having to go forward too fast. Other things that the duodenum will release are your enterogastrones, CCK, DLP1, and PYY. Those will inhibit gastric emptying, but CCK also will stimulate the secretion of HCl. In the intestines, you also have some digestion there, especially in the jejunum. So before I get more into that, I'm going to talk about your anatomy of your duodenum, jejunum, and your ileum. Your small intestine, you have simple columnar cells, and they will have microvilli on them. Your lamina propria of your small intestines will also have areas in them called lacteals. So I've drawn a representation to the right of the small intestine, like layers that I have, where you have your lacteals inside of your lamina propria. You also have two layers of muscles, your inner circular and outer longitudinal for your muscularis externa, and then you have a mesenteric plexus that line between. And again, I think that contributes to the segmentation or the movements of peristalsis of the intestines to move through. It's in particular for the small intestines, you have crypts of Lieberkühn, and so those are where your stem cells and Paneth cells live. Your Paneth cells are there to help protect your stem cells, and your stem cells will grow from the bottom up, and your cells in your intestine have about a 2 to 5 day lifespan. And so at the very top, you'll have a little tip at the plica circularis, which are like the folding indentations inside the small intestine, where you have the epithelium slough off. In particular for the duodenum, your plica circularis are shorter in relation to your jejunum, which makes sense because if you have more of the plica, you have more surface area, more contact with the chyme and digestion and absorption. And then in your ileum, which is the last part, you do not have many or any, if at all, plica circularis because you're not doing as much absorption or digestion there. In particular, the one type of cell that you do have in the ileum of your intestine is Peyer's patches, which will secrete immunoglobulin A, most common immunoglobulin secreted in the body, lines mucous membranes, and will trap bacteria on them. Okay, going back to the duodenum, sorry, I'm jumping around. You have two different types of secretory cells in there as well. You have succus entericus, which will secrete salt and mucus. Then you also have Brunner's glands, which will secrete alkaline mucus. Again, another way to protect your duodenum and the rest of your intestines from being digested. In particular, again, going back to CCK, you have pancreatic juices that will be released into your stomach. And so the CCK will signal that to your pancreatic cells. I will get into those cells later, but going to what is released, you have your juices that go into your ampulla vater, goes into the sphincter of Oddi. The CCK also causes the sphincter of Oddi to contract. And then as for another contraction, you have gallbladder, which stores bile. CCK will stimulate the bile smooth muscle to contract and secrete bile into your sphincter, into your levator and sphincter of Oddi for fat absorption and digestion. Let's see what else am I missing. I'll talk about liver and gallbladder later. Okay. Any questions or anything that you saw I missed on here before I move on to the jejunum? Jejunum, you have brush border enzymes on the tip of your cells. What do? They digest your oligosaccharides, proteins with your aminopeptidases. They also have enterokinase, which is used to activate your pancreatic juices, which again, stimulated by CCK. So pancreatic juices, as they're secreted into the levator, sphincter of Oddi. You have pancreatic lipase and amylase to finish digesting fat and carbs. You have trypsinogen, which is converted to trypsin by enterokinase. And then also trypsin is autocatalytic, so it can also convert trypsinogen to make more trypsin. What does trypsin do? It does two things. It can digest and degrade basic amino acid residues, but it also converts chymotrypsin and procarboxypeptidase to their active forms. Chymotrypsinogen, active forms chymotrypsin that degrades your aromatic residues and also what's that? Glutamate, leucine, and methionine as well. Procarboxypeptidase, active form is carboxypeptidase. You have two isoforms of that, A and B. A is for your aromatic C-terminus of your residues and also your neutral residues. And then B is for your basic C-terminus residues. If anyone's going to ask about what breaks down acidic, unknown, kind of just assuming that pepsin might have a role in that and also maybe just digestion in stomach, but unconfirmed.
What do you mean by what breaks down acidic, like in an acidic environment?
Acidic residues. So like, because a bunch of these amino acids that these break down, basic, aromatic, neutral, we haven't really addressed too many acidic ones, unfortunately. And I have not found what addresses those. But if that's not from lectures, then I'm not going to worry too much about it. What's absorbed in the jejunum? You can absorb calcium through your TRPV6 and calcium ATPase. I will be revisiting that with parathyroid. You can also absorb sodium through your NHE and ATPase. And so you also have, if you have a sodium ATPase, you're always going to have potassium going in the opposite direction. So you also have your potassium chloride co-transporters, which will co-transport chloride and potassium. You'll also secrete bicarbonate into the lumen of the intestines as an exchange for the calcium, not calcium, chlorine, which makes sense because again, acidic environment. You also have carbs that are absorbed in your jejunum through SGLT1 and 2s and GLUT2 and 5. They're co-transported with salts. Your GLUT2s will do most of your hexakinases, glucose, galactose, whereas GLUT5 will in particular reabsorb fructools. Then you also have amino acid absorption, co-transported with sodium, and that should be all for the jejunum. For the ileum, which I forgot to draw the name of because I erased it underneath the peristalsis, ileum. You are reabsorbing your bile salts. And then in particular, your bile salts being reabsorbed will trigger your liver to release more bile salts, which kind of makes sense because if you're releasing bile salts early on to digest, you want to make sure you can absorb everything you can, so release more. It's the most potent stimulator of the releasing of bile salts into the sphincter of Oddi. You also have vagal inputs that will also stimulate bile salt release, and this is namely in an anticipatory manner. So you see a food you like, you want to eat it, so might as well get ready to digest it because you're probably going to eat it unless you have really good self-control. I wish I had that. You have vitamin A reabsorption in the ileum. You also have in the terminal ileum, particularly B12 and intrinsic factor absorption. Intrinsic factor binds to B12 and it's required for the absorption. You go through the receptor QBAM and then that complex of B12 and IF is endocytosed. And then you do, okay, and actually, no, I'll just leave it at that. Let me think of anything I'm missing for intestines. Also, as a side note that I kind of forgot about, for gastric emptying, you have two other things that'll slow it down. You have emotional damage and pain. And so those would be mostly sympathetic, which makes sense. You're not trying to rest and digest. Emotional damage. In large intestines.
Poop.
Okay, poop, yes. In large intestines, you have, for anatomy, you have your mucosa, it's now flat columnar, no villi. You're not doing as much you don't need as much surface area as the small intestine. Your submucosa, in particular, will have lymphoid tissues. In your muscularis externa, you have two layers, your inner circular and your tinea coli, and that's also known as your outer longitudinal. They run longitudinally along the intestines, and you can see this grossly, I think, in anatomy, or I think someone did, showed it to me. In the serotian and adventitia layer, which is deepest to the muscularis externa, you also have your appendices epiloicae, and someone made a comment on that. This is anatomy, this is a physiology board, so I will ignore that and let you guys review that on your own, because I don't know it. There's still a mild amount of digestion going on in your large intestine, although it's mostly done by bacteria, not by enzymes that your body's making. So your bacteria can digest any leftover carbs into methane, and then you also produce H2S, which is the smelliness of your farts. You also make vitamin K, B complex, and folic acid from your bacteria. Otherwise, in your large intestine, what you're mostly absorbing is your water, your salts, electrolytes, to consolidate your stool. In your small intestines, like I mentioned, you have segmentation. In your excuse me, large intestine, you have your hauscal contractions. You also have something called mass action contractions, which only occur in the ascending and transverse parts of your intestine, and that is stimulated by gastric release, which is stimulated by eating. Think of what else is there for the intestine. I think that is all for the large intestine. Going to briefly talk about the liver since I skipped over it. I talked mostly about the gallbladder a little bit. In your liver, you have, for histology, just to be complete, your hepatocytes, which are kind of like hexagonal shaped from what was represented to us in our lectures. You have different divisions of, or different ways of dividing your histology. You have your classic lobule, which is just your single hepatocyte. You have your pleural lobule, which is dictated by where your or your bile drains to, excuse me. And so that's represented kind of in a triangle where that bile duct is. And then you have your hepatic ancinus, which is dividing your hepatic cells by proximity to your hepatic arterioles. And so closer to the hepatic arterioles, you have more oxygenation. As you get further out, closer to the center of the hepatocytes, you have less oxygenation. This also has to do with exposure to toxins. So, you know, you're exposed more to toxins at the periphery of each individual hepatocytes than closer in. You also have, as you zoom out into like kind of a unit of your hepatocytes, they're all surrounding a central vein, which goes to an interlobular vein. Each unit is kind of surrounded by a space called the space of Dis, where you have two different types of cells, stellate cells and Kupffer cells. Stellate cells will store your vitamin A, but it also is what causes your liver to be fibrotic in cirrhosis. So it'll synthesize collagen when you have liver damage. Your Kupffer cells are your macrophages, the immuno part of your liver. This is where bile secreted. Gallbladder again stores your bile salts. I will move on next to. I think I'll move on next to the pancreas actually before I move on to the rest of endocrine, because that one seems to be a little more separated out and also relates more to the stomach. So your pancreas, you have two different secretions to it. You have your endocrine secretions and your exocrine. Your exocrine will have these ducts histologically. At the end of the ducts are your acinar cells, and they have granules in them because if you're secreting stuff, you're gonna have granules to release stuff. That will the duct are lined by flattened columnar cells, and then that transitions into columnar cells, or did I say columnar? Flattened cuboidal cells transition to columnar. So you go from your intercalated cells, your intercalated duct, to your interlobular, then to your common bile duct, where you release into your epilevator and sphincter of Oddi. There is some recycling that happens between your parietal cells and your exocrine cells of the parietal cells of the stomach, exocrine cells of the pancreas. So how do you make hydrochloric acid? Well, you have salt in your body. Salt is NaCl. You also have water and carbon dioxide. Carbon dioxide and water make bicarbonate, and it also makes proton as a byproduct. So then you have HCl from your salt and the production of bicarbonate. So that is what is going to be secreted into the stomach. So what do you do with that bicarbonate that's left over then? You can use that to make NaHCO3, which is again used as the buffer in your intestines. And so you combine that with the Na that's left over from your salt, and that's also secreted in your pancreatic juices. And that again, just to reiterate, stimulated by secrete. I also have, as a side note, secretin inhibits gastric emptying, our favorite thing, which makes sense because if you're secreting that, that means you have food in your intestines, so you want to slow down gastric emptying. Moving on from the exocrine part of the pancreas to the endocrine part of the pancreas. Your cells are organized in islets of Langerhans. You have different types of cells in your endocrine system. For the pancreas, you have your alpha cells, which will release glucagon, your sigma, which will release somatostatin, again, more inhibitory effects. And then you have epsilon cells, which will release ghrelin. And then your beta cells, which will release insulin. To make insulin, you have proinsulin, which is in the endoplasmic reticulum of the beta cells. And then eventually you use PSCK1 and 2 to cleave off a C-peptide from the proinsulin. And then you also have insulin as a result. As a note, C-peptide is often used in diagnosing type 1 versus type 2 diabetes, because in type 2 diabetes, you're still making it. Whereas in type 1, you're not making any insulin at all, so you don't have any C-peptide. And C-peptide is measured because it has a longer half-life than insulin, so easier to measure than insulin itself. So once you make insulin, what do? Insulin binds to tyrosine kinase. Tyrosine kinase will signal to IRS, and that will have different effects depending on the cells you have in location to wherever in the body. I'm not going to get into those specifics. I'll just mention the different pathways. So if your IRS can signal to a PDK, which will eventually increase expression of GLUT4, excuse me, which is your insulin-dependent glucose transporters. Those are expressed in skeletal muscles, for example. Then you can also signal to G proteins. So GS will increase your glycogen synthase, which again makes sense if you're increasing glucose, you're wanting to use it as much as you can. You can use it for energy, or you can use it for storage. Glycogen synthase makes the glycogen for storing. Then you can also signal mTOR. mTOR is usually signaling, protein synthesis. Again, if you are not trying to use protein for energy, might as well build it up. So liver in particular, I think, is one. Liver and then the ribosomes of skeletal muscle will do protein synthesis, I think. What else does insulin stimulate? Oh, actually, I was starting to write rapid, intermediate, and long-term effects of insulin, and I forgot to finish it. But just to briefly recap, rapid happens within seconds to minutes. You'll increase transport of glucose, which is again your GLUT4 is your independent glucose dependent insulin dependent glucose transporter. You also transport amino acids and potassium as well. Your intermediate effects, I believe, take a few more minutes. I'm gonna just look at my notes really quick because I forgot to write this. Unless someone knows what the intermediate effects are for insulin. Yes. You have until I get to my notes. Oh, spaghetti notes. Okay, so intermediate is minutes. You have increased protein synthesis, again, mTOR pathway. Decreased protein degradation, because again, you're not trying to use protein for energy. You have more glycolytic enzymes, so again, more glycogen synthase and less phosphorylase, which is for your gluconeogenic enzymes. Your delayed effect, which takes several hours, increased mRNA for lipogenic and other enzymes. Let me think what else does insulin. So then you also have regulation of insulin. Starting with glucagon, this one is a kind of a paradoxical one. Glucagon is released in your fasting state. It will activate GPCRs, and stimulate calcium release from beta cells and actually, in a sense, increase the release of insulin. So why does that make sense if they're kind of working against each other? My thought was that glucagon, you're trying to stimulate the liver to release glucose, right? For other muscles. And so you need at least a little bit of insulin to help, your GLUT4s say, hey, there's some insulin. Let's take some in. That's my thought, neither confirmed nor denied. What else can cause release of insulin? You can have amino acids and keto acids inside of your blood that will close your ATP potassium channels. What that does when you close those channels, it depolarizes your beta cells. With your depolarization, you increase your calcium levels in the cells, which again releases insulin. You can also have ACH stimulation of the M3R receptor, calcium to insulin. You also have our most classic pathway, I believe, GSIS or glucose stimulated insulin secretion. Glucose goes to GLUT2. GLUT2 will increase calcium levels and therefore calcium insulin again. In regards to what can actually inhibit insulin release, somatostatin, our favorite. It will signal a G inhibitor protein, which will decrease levels of CAMP and that will inhibit insulin secretion. You can also have sulfonylureas, which will prevent the closing of the ATPK channels, which will not depolarize it, will not release insulin. And then one more thing. If you have release of
I thought sulfonylurea is the one that is going to increase insulin, at least because it's a drug, right?
actually, hold on.
Yeah.
Did I write that wrong then? Oopsies. So then it's increased. My bad. This is why I have you guys smarter than me. Sulfonylureas will stimulate ATPK channel closure and therefore release more insulin. That makes more sense because it's a diabetic drug. My bad. Yeah, no, thank you. Where was I? Norepinephrine and epinephrine released in sympathetics. You are not trying to rest and digest. Alpha 2, it will be stimulated, which is a GI, again, reduced levels of CAMP, inhibit insulin, because you're not trying to rest and digest. I'm trying to see. I think that is everything for the pancreas. So now I will move on to the rest of the endocrine system. So starting with hypothalamus, you have somatostatin, again, more inhibitory effects, CRH and TRH, which will be involved in cortisol and your thyroid respectively. So let's start with cortisol, why not? So CRH will be released and that binds to CRH1R receptor, which will increase your adenyl cyclase, increase CAMP, my favorite cascade, and that will increase release of ACTH from the pituitary gland. Actually, let me talk more about the histology of the pituitary as well. Hypothalamus, you have infundibulum systolic to the pituitary. Your pituitary is divided into your anterior and posterior. And then you also have cells in the middle, which are not as important, but the cells in the middle are your pars intermedia. If you're looking on a histological slide, they'll have colloid. That's the only way to identify it. Function is unknown. And posteriorly, you have your pars nervosa cells, which will release oxytocin and ADH. And then you have your pars distalis on anterior, which will have 3 different cell types. Your basophils, which stain because they like bases, acidophils, which stain red. Basophils are blue, acidophils are red, and then you have ones that don't stain, but we are not concerned with those here. Your basophils will have your gonadotropes, which will make your luteinizing hormone and FSH. Your thyrotropes are also basophils, which will release thyroid stimulating hormone in a pulsatile manner. And your corticotropes, which will release ACTH, which we just mentioned. In regards to some of the hormones released by the basophils, luteinizing hormone, FSH, and TSH all share a similar or the same alpha subunit, but the beta subunit is different. So that's what stimulates the different effects. ACTH. Let's go to adrenal glands now. Your adrenal glands. You have your cortex, which includes your zona glomerulosa, zona fasciculata, and zona reticularis. You have your medulla, which include your chromatin cells. Histologically, your glomerulosa releases mineralocorticoids, which are your aldosterone. There will be like larger cells with fat deposits in them, foamy fat deposits. Your fasciculata releases your glucocorticoid. And androgens, they are arranged more in like paired columns, and they also have foamy fat cells in them, or fat filled, lipid filled accumulations. Question? I have that fasciculata releases androgens, but then earlier today, someone pointed out to me that DHEA is also an androgen. So I guess both kind of do androgens, or maybe just focus.
Do primarily reticularis androgens.
Yeah, okay. So then
I think
reticular.
it's fine.
Okay, so fasciculata and reticularis both do androgens. Androgens are mostly for reticularis. Reticularis cells don't have the foamy fats of the glomerulosa and fasciculata, but they do have granules in them, and they're arranged in a single row usually. So how do we make ACTH, or how do we make cortisol from ACTH? ACTH will bind in the fasciculata, which makes your glucocorticoids, will bind to your MCR2 receptor. Where is it? MCR2 receptor, which will start the cascade of adenyl cyclase that camp to PKA. PKA will activate your star. What's your star? It's the star of the show. It's the rate limiting aspect of cortisol synthesis. It will, when you activate it, it lets cholesterol transport into the mitochondria. And once you have the cholesterol in the mitochondria, you can undergo reactions to make cortisol. You have different CYPs that we want to talk about here. Cholesterol is made to progenolone by CYP1A1 or 11A1. I'll just say the numbers instead of CYP to be less redundant. So 11A1 makes progenolone. The 3-beta HSD11 will convert progenolone to progesterone. It will also convert. You can use CYP17 to convert the progenolone and progesterone to the 17 hydroxy forms of it. And so the 17 hydroxy forms are what make the cortisol on the left side of this reaction here, on the right side of the reactants. From the progesterone, you use 21 and 11B1 to make cortisol or corticosterone, depending on which side you're on. Once you make your cortisol, you have your HPA axis. It will signal back and inhibit the release of ACTH and CRH. So that's your hypopituitary axis, adrenal axis, I mean, right there. I'm not going to go into the effects of cortisol. Those are longer term stress effects. I did not have room to write those on here, so I do not know them, unfortunately. What else does your adrenal glands make? Going to your reticularis actually. It kind of hijacks a lot of the same proteins making cortisol, except you use CYP17 again to make DHEA. For glomerulosa, where you're making aldosterone, again, you're using cholesterol as your progenitor molecule. You have, if you remember FOM4, angiotensin 2 activating angiotensin 1 receptor, which increases DAG, increases PKC, which will cause synthesis of cholesterol to progenolone. Then you use CYP11B2 instead of B1 to make aldosterone, and then aldosterone will do its various effects. I think where I want to go from here. Okay, chromatin cells in the medulla. you're making epi, norepinephrine, your catecholamines. Dopamine is also catecholamine. These will have a pinpoint nucleus histologically and will also have granules. How do you make epi and norepinephrine? Use tyrosine. You use your enzyme PNMT to make epinephrine. And so that's, easy enough, epinephrine. And then you can use that to signal to, again, alpha 2 receptors or wherever else for sympathetics. You also use COMT to convert epi to metanephrine. And then you can make norepinephrine, from metanephrine. And then you can also make DHPG with MAO from NAG and then COMT again to normetanephrine. I think those are less important. How do you stimulate release of catecholamines? This is something that's important that they drilled into us. Acetylcholine will stimulate nicotinic receptors to release catecholamines. Why is acetylcholine if it's parasympathetic related most of the time? Well, these are preganglionic cells, and so the preganglionics will always deal with acetylcholine. And then what's the ganglion of the adrenal gland? Medulla cells themselves. And if you want to think about it embryologically, neural crest cells, there are what the medulla stem from. So it makes sense that they're very neural-like. It is its own ganglion. So now we have finished cortisol, ACTH. Cortisol also will affect TSH, so I will go to TSH right now for your thyroid. TSH is released, is first released once you trigger it with TRH. TRH is made from pro-TRH in the hypothalamus. Once TRH is made, it binds to TRHT, which is a GQ GPCR, increases calcium levels and releases TSH from the thyrotropes. TSH will bind to TSHR in thyroid cells. Histologically, your thyroid cells are surrounding colloid, and the cells are called your follicular epithelium. So the follicular epithelium of TSHR binding to LC to DAG, or no, stimulate PLC to make DAG IP3, which will release thyroglobulin into your colloid. Thyroglobulin is used to make your T3 and T4. TSHR also activates adenylocyclase camp that will upregulate your NIS, which is your co-transporter of sodium and iodine, or iodide. Iodide is used to make T3, so iodide is transported into your cell, excreted into the colloid by pendrin. The thyroglobulin is combined with your iodide, and then you react that with TPO and peroxides, which is made by TXO2, to make TG-MIT or TG and DIT, or thiglobulin MIT and DIT. And so you combine two of those. So depending on whether you combine MIT and a DIT for T3 or two DITs for T4, you use TPO again, and then you transport that complex of thiroglobulin T3, T4 into your follicular epithelium. You'll use a protease or lysosomes to cleave off the T3 and T4 and secrete those out. You can also use D1 and D2 to convert your T3, 4 into T3. Your T4 and T3, once they're released, there's most of them are bound, 99% of them. As they travel in the blood, they will be bound to thyroid binding globulin, TBG, which is your major transporter. You'll also have TBPA and albumin, which are more minor. I believe albumin I was told, which makes sense, is low affinity but high in concentration, whereas TBG is low in concentration levels but very high affinity. Thyroid hormone, some of its effects, you have your 6Bs, basal metabolic rate, increases UCP and mitochondria, which will increase the heat generation. You have increased breakdown, which is increased glycogenolysis, increased blood glucose, which is from your glycogenolysis, and increased uptake. And then increased beta adrenergic stimulation, because you increase the number of receptors and channels open. And then also increasing phospholambda, which, if you refer to FOM4, increases contractility of your heart. Also affects brain development and function, and then bone development and function as well. In regards to blood glucose, thyroid hormone will increase your absorption of carbs in your small intestine in the jejunum by increasing the amount of SGLT1/2s and GLUTs. If you have hyperthyroidism and hypothyroidism, those affects your 6Bs. So again, hyper, you have too much, you have more basal metabolic rate, more body heat, more breakdown of stuff. So not just carbs, but also protein, so you can get muscle weakness. You have more beta adrenergic stimulation, so faster heart rate, anxiety with that, increased bowel movements, and then also more nervous tremors, and brisk reflexes. Hypothyroidism is the opposite, lower basal rate, lower body heat. You also of note increase your total and LDL cholesterols. You have constipation, and then you decrease your beta adrenergic, so you can have bradycardia, and then you have fatigue and lethargy. So also once you release, as a note, if you have excess iodine, it can have two different effects on making T3 and T4. You can either have what's known as like the Wolff-Raynaud effect, where you decrease production by inhibiting pendrin, so therefore inhibiting import of iodine into the colloid, so less production of T3, T4, or you inhibit TPO, which is the big T3, T4 creator in the colloid as well. You can also have the Jod-Basedow effect, which just means you have increased T3, T4 from excess iodine, which kind of makes sense if you have more of one thing, you can make more of the rest. I think that is all for thyroid. T3, T4 signal back to inhibit TSH and TRH release. Other things that inhibit TSH release are cortisol and somatostatin from the hypothalamus. And I believe that should be all that inhibited. Cool. Let's talk about glucocorticoids. Yeah, cortisol. Dopamine. Oh, and dopamine. Thank you. Yeah, and dopamine also inhibits the release of TSH, thyrotropes. I think I'm almost done. I just have parathyroid left. Perfect. Parathyroid. Your cells are much smaller in your parathyroid, and histologically they'll appear more dense, with larger nucleus in relation to the size of the cells, I believe. What is parathyroid good for? You ask. Calcium regulation is its thing. So so if you have, let's say, low calcium levels and you want to increase them, you will increase secretion of PTH. PTH will signal to PTHR1, 2, and 3. The 1 is the one we care about because it signals to bones and kidneys. How does it signal? It will increase your cyclic adenylyl cyclase. That is the wrong way. That is AC, not CA. My bad. Adenylyl cyclase will increase your levels of CAMP, increase pKa, and what that does, particularly in the kidney, is increases your TPRV5. Oh, I don't remember how it goes. ECAC increases ECAC in the distal tubule, and then decreases NAPI or phosphate transport in the proximal tubule. So you'll have that difference of increasing calcium and decreasing phosphate in your serum, or vice versa in your urine. You can also increase in terms of your bones, rank L production, which are expression on the just rank L expression, which will activate rank, which is receptor on your osteoclast, and so that will stimulate your osteoclast to break down bone, release more calcium and phosphates. You also have stimulation of activation of vitamin D. So vitamin D is activated from its inactive form, which is created in the liver by Morgan, from 25-OH-D3 into its active form of 1,25-OH-2D, which happens in the kidney. What that does, it will actually enhance calcium absorption in the intestine by increasing expression of TPRV6 and CA-ATPase in the intestine. It also stimulates both osteoclasts and osteoblasts in bone remodeling. Which makes sense that it stimulates both because you need both for bone remodeling. For good healthy bones. What else do I want to go? For inhibition of PTH, as your calcium levels rise to about greater than 9 milligrams per deciliter, your C cells, your polyfollicular cells in your thyroid will release calcitonin to inhibit the secretion of PTH. Yeah.
Sorry, can I make a quick note?
Yeah.
For bone remodeling, when PTH has to go through osteoblast first, before it gets to osteoclast.
It goes,
That's like a kind of protective mechanism, so that we're not just breaking down bone.
okay.
So,
So PTH does
yeah, right,
directly.
it directly goes to osteoblast, which has the rank L.
Okay.
Which then binds to rank, which is on the osteoclast.
Okay, so it stimulates both.
Well, it's, directly, PTH directly in rank L,
Yeah, directly on rank L, which is
on
at
the
osteoblast.
osteoblast.
Okay.
Yeah.
So I'll make that note.
I don't know how important it is, but I just.
No, it's good. Thank you.
Protective thing.
Yeah, that makes sense. You want to build up before you break down this for like a fucker.
Yeah.
Okay. I learned something new. Literally 5 things today. Calcium levels will release calcitonin. There's also CASR, which will lead it to a cascade that will degrade PTH. So your cascade is PKC, using phospholipase 2 to an arachidonic acid cascade to degrade PTH. That is also stimulated, if PLA2 or the phospholipase is also stimulated by phosphate, which is released from bone. And then your vitamin D will also inhibit release of PTH through VDR. And then also phosphate will also inhibit the creation of vitamin D. And then you also have FGF 23, which will also inhibit the 1-alpha hydroxylase, which will decrease vitamin D level creation. And
Where
wow.
are the VDR and FGF?
I do not know, unfortunately. There's just side notes that I had. I think those are the important parts. I just wrap this up with saying that you, with your prolactin, which is created by like lactotropes, it is always on. It's stimulated by estrogen, and then otherwise it can be inhibited by somatostatin. It can also be inhibited by dopamine. It's stimulated by both ADH and oxytocin, which is kind of interesting. Somatotropes have growth hormone releasing hormone and then growth hormone. GHRH will stimulate growth hormone, which will stimulate SS, which will inhibit GHRH for a very simple negative feedback. Prolactin will also inhibit GHRH. And then GHRH is also stimulated by ghrelin, which is interesting because if you're hungry, why would you want to like stimulate growth hormone for growing? But that's something I haven't figured out. And I think that is all of it. Oh boy. Anything I missed and any other questions? All zoned out? That's really good. I'm gonna go